Welcome to the certificate course on reproductive health education. Today's session, let us continue with the male reproductive system. We will be speaking about testes. Inside the seminiferous tubules are six different cells. These include supporting cells called succenticular cells, as well as five types of developing sperm cells called germ cells. Germ cell development progresses from the basement membrane at the perimeter of the tubule toward the lumen. Let's look more closely at these cell types. Serotoli cell. Surrounding all stages of the developing sperm cells are elongate branching serotoli cells. Serotoli cells are a type of supporting cells called succenticular cells or sustenocyte that are typically found in epithelial tissue. Serotoli cells secrete signaling molecules that promote sperm production and can control whether germ cells live or die. They extend physically around the germ cells from the peripheral basement membrane of the seminiferous tubules to the lumen. Tight junctions between these sustentacular cells create the blood-testis barrier, which keeps the blood-borne substances from reaching the germ cells and at the same time keeps surface antigens on developing germ cells from escaping into the bloodstream and prompting an autoimmune response. Germ cells Germ cells, the least mature cells, the spermatogonia, line the basement membrane inside the tubule. Spermatogonia are the stem cells of the testes, which means that they are still able to differentiate in a variety of different cell types throughout adulthood. Spermatogonia divide to produce primary and secondary spermatocytes, then spermatids, which finally produce formed sperm. The process that begins with spermatogonia and concludes with the production of sperm is called spermatogenesis. Spermatogenesis. As just noted, spermatogenesis occur in the seminiferous tubules that form the bulk of each testis. The process begins at puberty, after which time sperms are produced constantly throughout a man's life. One production cycle from spermatogonia through formed sperm takes approximately 64 days. A new cycle starts approximately every 16 days, although this timing is not synchronous across the seminiferous tubules. Sperm count, the total number of sperm a man produces, slowly decline after age 35, and some studies suggested that smoking can lower sperm counts irrespective of age. The process of spermatogenesis begins with the mitosis of the diploid spermatogonia. Because these cells are diploid, they each have a complete copy of the father's genetic material or 46 chromosomes. However, mature gametes are haploid, containing 23 chromosomes, meaning that daughter cells of spermatogonia must undergo a second cellular division through the process of meiosis. Spermatogenesis. Mitosis of a spermatogonial stem cell involves a single cell division that results in two identical diploid daughter cells, spermatogonia to primary spermatocyte. Meiosis has two rounds of cell division, primarily spermatocyte to secondary spermatocyte and then secondary spermatocyte to spermatid. This produces four haploid daughter cells, that is spermatids. In this, in this electron micrograph of a cross section of a seminiferous tubule, the lumen is the light shaded area in the center of the image. The location of the primary spermatocyte is near the basement membrane and the early spermatids are approaching the lumen. Two identical diploid cells result from spermatogonia mitosis. One of these cells remains a spermatogonium and the other becomes a primary spermatocyte. The next stage in the process of spermatogenesis. As in mitosis, DNA is replicated in a primary spermatocyte and the cell undergoes cell division to produce two cells with identical chromosomes. Each of these is a secondary spermatocyte. Now, a second round of cell division occurs in both of the secondary spermatocytes, separating the chromosome pairs. This second meiotic division results in a total of four cells with only half of the number of chromosomes. Each of these new cells is a spermatid, although haploid early spermatids look very similar to cells in the earlier stages of spermatogenesis with a round shaped central nucleus and large amount of cytoplasm. A process called spermiogenesis transforms these early spermatids, reducing the cytoplasm and beginning the formation of the parts of the true sperm. The fifth stage of germ cell formation, spermatozoa or formed sperm, is the end result of this process, which occurs in the portion of the tubule nearest to the lumen. Eventually, the sperm are released into the lumen and are moved along a series of ducts in the testes toward a structure called the epididymis for the next step of sperm maturation. Structure of sperm. Sperm are smaller than most of cells in the body. In fact, the volume of a sperm cell is 85,000 times less than that of the female gamete. Approximately 100 to 300 million sperms are produced each day, whereas women typically ovulate only one oocyte per month. As is true for most of cells in the body, the structure of sperm cells speaks to their function. Sperm have a distinctive head, midpiece, and tail region. The head of the sperm contains the extremely compact haploid nucleus with very little cytoplasm. These qualities contribute to the overall small size of the sperm. A structure called acrosome covers most of the head of the sperm cell as a cap that is filled with the lysosomal enzyme important for preparing sperm to participate in fertilization. Tightly packed mitochondria fill the midpiece of the sperm. ATP produced by these mitochondria will power the flagellum, which extend from the neck and the midpiece through the tail of the sperm, enabling it to move the entire sperm cell. The central strand of the flagellum, the axial filament, is formed from one centriole inside the maturing sperm cell during the final stages of spermatogenesis. Sperm cells are divided into a head containing DNA, a midpiece containing mitochondria, and a tail providing motility. The acrosome is oval and somewhat flattened. Sperm transport. To fertilize an egg, sperm must be moved from the seminiferous tubules in the testes through the epididymis and later during ejaculation along the length of the penis and out into the female reproductive tract. Role of epididymis. From the lumen of the seminiferous tubules, from the lumen of the seminiferous tubules, the immotile sperm are surrounded by a testicular fluid and moved to the epididymis, a coiled tube attached to the testes where newly formed sperm continues to mature. Though the epididymis does not take up much room in its tightly coiled state, it would be approximately 6 meters or 20 feet long if straightened. It takes an average of 12 days for sperm to move through the coils of the epididymis, with the shortest recorded transit time in human being 1 day. Sperm enter the head of the epididymis and are moved along predominantly by the contraction of smooth muscles lining the epididymal tubes. As they are moved along the length of the epididymis, the sperm further mature and acquire the ability to move under their own power. Once inside the female reproductive tract, they will use this ability to move independently toward the unfertilized egg. The more mature sperm are then stored in the tail of the epididymis until ejaculation occurs. Duct system. During ejaculation, sperm exit the tail of the epididymis and are pushed by smooth muscle contraction to the ductus deferens, also called the vasa deferens. The ductus deferens is a thick muscular tube that is bundled together inside the scrotum with connective tissue, blood vessels, and nerves into the structure called the spermatic cord. Because the ductus deferens is physically accessible within the scrotum, surgical sterilization to interrupt sperm delivery can be performed by cutting and sealing a small section of the ductus deferens. This procedure is called a vasectomy and it is an effective form of male birth control. Although it may be possible to reserve a vasectomy, clinic clinicians consider the procedure permanent and advise men to undergo it only if they are certain they no longer wish to father children. From each epididymis, each ductus deferens extends superiorly into the abdominal cavity through the inguinal canal in the abdominal wall. From here, the ductus deferens continues posteriorly to the pelvic cavity, ending posterior to the bladder where it dilates in a region called the ampulla. Sperm make up only 5% of the final volume of semen, the thick milky fluid that the male ejaculates. The bulk of semen is produced by three critical accessory glands of the male reproductive system: the seminal vesicle, the prostate, and the bulbourethral glands. Seminal vesicle. As sperm pass through the ampullae of the duct deferens at ejaculation, they mix with the fluid from the associated seminal vesicle. The paired seminal vesicle are glands that contribute approximately 60% of the semen volume. Seminal vesicle fluid contains a large amount of fructose, which is used by the sperm mitochondria to generate ATP to allow movement through the female reproductive tract. The fluid, now containing both sperm and seminal vesicle secretions, next move into the associated ejaculatory duct, short structure formed from the ampulla of the ductus deferens and the ductus of the seminal vesicle. The paired ejaculatory ducts transport the seminal fluid into the next structure and the prostate gland. Prostate gland. The centrally located prostate gland sits anterior to the rectum at the base of the bladder surrounding the prostatic urethra, the portion of the urethra that runs within the prostate. About the size of the walnut, the prostate is formed of both muscular and glandular tissues. It excretes an alkaline milky fluid to the passing seminal fluid. We can call it as a semen that is critical to first coagulate and then decoagulate the semen following ejaculation. The temporary thickening of the semen helps retain it within the female reproductive tract, providing time for sperm to utilize the fructus provided by seminal vesicle secretions. When the semen regains its fluid state, sperm can then pass further into the female reproductive tract. Bulbourethral glands. It will add to semen. So the final addition to semen is made by these corpus glands or bulbourethral glands. It releases thicky salty fluid that lubricates the end of the urethra and the vagina. Next, hormones. Uh, Hormones are testosterone. It is an androgen, is a steroid hormone produced by Leydig cells. The alternate term for Leydig cells, interstitial cells, reflects their location between the seminiferous tubules in the testes. In male embryos, testosterone is secreted by Leydig cells by the seventh week of development, with a peak concentration reached in the second trimester. This early release of testosterone result in the anatomical differentiation of the male sexual organs. In childhood, testosterone concentrations are low. They increase during puberty, activating characteristic physical changes and initiating spermatogenesis. Functions of testosterone. The continued presence of testosterone is necessary to keep the male reproductive system working properly, and Leydig cells produce approximately 6 to 7 milligram of testosterone per day. Testicular steroido, steroidogenesis result in testosterone concentrations that are 100 times higher in the testes than in the circulation. Maintaining these normal concentration of testosterone promotes spermatogenesis, whereas low levels of testosterone can lead to infertility. In addition to intratesticular secretion, testosterone is also released into the systematic circulation and plays an important role in muscle development, bone growth, and development of secondary sexual characteristics and maintain libido, that is, sex drive, in both males and females. In females, the ovaries secrete small amount of testosterone, although most is converted to estradiol. A small amount of testosterone is also secreted by the adrenal glands in both sexes. Control of testosterone. The regulation of testosterone concentration throughout the body is critical for male reproductive function. The intricate interplay between the endocrine system and the reproductive system is shown in this particular diagram. So, here we could see the regulation of testosterone production. The hypothalamus and the pituitary gland regulate the production of testosterone and the cells that assist in spermatogenesis. Gonadotrophic hormones activates the anterior pituitary to produce luteinizing hormone and follicle stimulating hormone, which in turn stimulate Leydig cell and serotonin cells respectively. The system is a negative feedback loop between the regulation of Leydig cell production of testosterone begins outside the testes. The hypothalamus and the pituitary gland in the brain integrate external and internal signals to control testosterone synthesis and secretion. The regulation begins in the hypothalamus. Pulsatile release of a hormone called gonadotropin releasing hormone from the hypothalamus stimulates the endocrine release of hormones from the pituitary gland. Binding of gonadotrophic hormone to its receptor on the anterior pituitary gland stimulate release of the two gonadotropins, that is luteinizing hormone and follicle stimulating hormone. These two hormones are critical for reproductive function in both men and women. In men, follicle stimulating hormone binds predominantly to the serotonin cell within the seminiferous tubules to promote spermatogenesis. Follicle stimulating hormone also stimulate the serotonin cells to produce hormones called inhibins, which function to inhibit follicle stimulating hormone release from the pituitary, thus reducing testosterone secretion. These polypeptide hormones correlate directly with the serotonin cell functions and sperm number. Inhibin B can be used as a marker of spermatogenic activity. In men, luteinizing hormone binds to receptor on Leydig cells in the testes and upregulates the production of testosterone. A negative feedback loop predominantly controls the synthesis and secretion of both follicle stimulating hormone and luteinizing hormone. Low blood concentration of testosterone stimulate the hypothalamic release of gonadotropin hormone. Gonadotropin hormone then stimulate gonadotropin releasing hormone, then stimulate the anterior pituitary to secrete luteinizing hormone into the bloodstream. In the testes, luteinizing hormone binds to luteinizing hormone receptor on Leydig cells and stimulate the release of testosterone. When concentration of testosterone in the blood reach a critical threshold, testosterone itself will bind the androgen receptors on both the hypothalamus and the anterior pituitary. inhibiting the synthesis and secretion of gonadotropin releasing hormone and luteinizing hormone respectively. When the blood concentration of testosterone once again declines, testosterone no longer interacts with the receptors to the same degree, and gonadotropin releasing hormone and luteinizing hormones are once again secreted, stimulating more testosterone production. This same process occurs with follicle stimulating hormone and inhibin to control spermatogenesis. Aging and the male reproductive system. Declines the leading cell's activity can occur in men beginning at 40 to 50 years of age. The resulting reduction in circulating testosterone concentration can lead to symptoms of andropause, also known as male menopause. While the reduction in sex steroid in men is akin to female menopause, there is no clear sign such as that lack of menstrual period to denote the initiation of andropause. Instead, men report feelings of fatigue, reduced muscle mass, depression, anxiety, irritability, loss of libido, and insomnia. A reduction in spermatogenesis resulting in lowered fertility is also reported, and sexual dysfunction can also be associated with andropausal symptoms. Whereas some researchers believe that certain aspects of andropause are difficult to tease apart from aging in general, testosterone replacement is sometimes prescribed to alleviate some symptoms. Recent studies have shown a benefit from androgen replacement therapy on the new onset of depression in elderly men. However, other studies caution against testosterone replacement for long term treatment of andropause symptoms, showing that high doses can sharply increase the risk of both heart disease and prostate cancer. With this, I conclude a topic on male reproductive system. See you in the next session.